When samples arrive at the testing lab, the package is scanned to show the system that it was received by the lab and initialed by the lab technician. Samples are removed from the transportation container and are placed in a heat bath at 95 degrees Celsius. After 30 minutes, the samples are removed and placed on ice. A lab technician does a visual inspection of samples to determine if there is enough volume and correct consistency for the robotic equipment, and are separated for manual or robotic plating. Samples are scanned individually and their location on the tray are logged and paired with a sampling plate. Samples that are too thick or not suitable for transfer with a robot are transferred into a testing plate manually. The plate is labeled with a batch number and plate number for tracking. As a lab technician draws a precise amount from sample tubes to be set into the testing plate. The unused sample is spot checked to verify that it was placed in the correct slot, and all samples are temporarily stored in case retesting is required. Samples are held in the lab for 48 hours before proper disposal in red biohazardous waste bin. For the robotic plating process, vials are scanned and placed into a matching location on a new tray. The caps are removed from the vials and the well tray is placed in the robotic transfer machine. The robot pulls samples from vials and places them in the sampling plate. The sampling plate is sealed to prevent contamination, and the remaining sample left in the vial is scanned before transfer and temporarily stored in case retesting is required. Samples are held in the lab for 48 hours before proper disposal in red biohazardous waste bin. The plate containing the transferred samples is placed on ice while the master mix is made. The master mix is a combination of reagents that allows detection of a positive or negative sample. A precise amount of the master mix is added to each well of 384 well PCR plate. Nuclease-free water is added to 2 wells of the 384 well PCR plate to act as a negative sample control. Sample plates are then transferred into the 384 well PCR plate by a technician. The 384 well PCR plate allows for more samples to be tested at a time, speeding the testing process. A positive reference material is added to 2 wells of the 384 well PCR plate, which will act as a positive sample control. The 384 well PCR plate is sealed and vortexed for proper mixing of sample and reagents. The full plate of samples is delivered to another technician who loads the samples into an analysis machine which will detect any positive or negative samples in the plate. Results are generated in approximately 1 hour after the plate is loaded to the analysis machine. Upon completion of the analysis, the technician reviews the results for accuracy and uploads results to the Shield, Illinois Laboratory Information System, also known as LIMS, and individual results are made available on individual accounts.